Hello and welcome back. We're going to now discuss gene regulation, and we're going to talk about gene regulation in eukaryotic cells. All right? So things are going to get a little more complex than when we talked about prokaryotic cells and the lac operon. Eukaryotic cells, we're going to have a lot more factors that come into play. All right? As far as the gene organization goes, you're going to have regions known as enhancers. And typically those enhancers are going to be upstream or towards the 5' end. So this would be our 5' end. This would be our 3' end of our DNA molecule, because what we're talking about here is we're still at the DNA level when we talk about genetic organization. The enhancers are upstream, all right? And there's a reason they need to be upstream, because things are going to kind of fold to come together to start transcription in eukaryotic cells, all right? So enhancers are something that are unique to eukaryotic cells. We're still going to have a promoter associated with eukaryotic gene transcription. Just like our prokaryotic cell, the RNA polymerase is going to bind at the promoter, okay? And then we've got whatever our gene is. Now, in terms of eukaryotic cells, the genes aren't organized into operons. So the organization, you may have a gene that, for example, is involved in eye color next to a gene that is involved in making pancreatic enzymes, okay? They aren't necessarily related, and they would have separate promoters. So we can hypothetically have another promoter down here. This would be our gene for eye color. This might be our gene for those pancreatic enzymes, okay? And depending on what cell we're in, that's going to dictate where the enhancers are going to tell the RNA polymerase it needs to bind. All right, so we've got enhancers. We still are going to have a promoter region, okay? These tend to be repetitive sequences, typically A's and T's, repeats of A's and T's in various combinations. But there are certainly some sequences that are unique which define what a promoter is. I'm not worried that you know those sequences. What I want you to know with the promoter, this is where RNA polymerase is going to bind, okay? And then we're going to have our gene, okay? We're going to have some other factors now that are going to come into play with eukaryotic cells. There are going to be a lot more proteins that are going to be associated with eukaryotic transcription as compared to prokaryotic transcription, okay? So what we've got for our proteins, okay, the one you're already familiar with, that's the RNA polymerase, okay? We are also going to have activators, and we are going to have proteins known as transcription factors. Very often you'll see that abbreviated TFs, okay? And really what we've got in the diagram in your book, the activator protein is there. The activator protein might be something that binds to our enhancers. And this is going to help bend the DNA in conjunction with our, let me draw that as a circle. Transcription factors. The activators are going to bind, they're going to help bend the DNA, and it's going to help recruit those transcription factors in. And that complex of proteins sitting there is going to help the RNA polymerase not necessarily know where to bind, because the promoter region is that sequence that tells the RNA polymerase where to bind, but it kind of keeps everything nice and tight together so that the RNA polymerase can transcribe the gene it's supposed to transcribe. Okay? So the key thing I want you to know here with transcription in eukaryotic cells, unlike prokaryotic cells, everything happened, I drew it out linear. Eukaryotic cells, there's a lot of DNA bending that ends up happening. So you've got regions that are far away from the gene, but those enhancers are going to help get that process of transcription started and maintained within our eukaryotic cells. Okay? So we've got a lot of other factors involved. The main ones I want you to be aware of, the activators. Activators are going to bind DNA. Transcription factors sometimes can bind DNA, sometimes they bind other proteins, all right? But transcription factors are also an important part in getting that complex of proteins where it needs to be on the eukaryotic gene so that you can get transcription to happen, okay? So that's eukaryotic transcription, and that's really kind of a real quick overview of it. There are some other things that can affect transcription in eukaryotic cells. Methylation, the addition of a CH3 group to the DNA, because if you have that additional chemical group there, that can interfere with this whole complex coming together. So methylation, typically if genes are highly methylated, they're usually off, okay? And that's why, as it interferes with the whole complex that we've got going on there with eukaryotic transcription, right? If you have any questions, please let me know.